All right, we're going to give this a whirl, taking notes this way off a video, and this way you can go back and forth if you need to on things you don't quite understand. I'm going to start with a little bit of a review here. We've got homozygous or heterozygous. An offspring will inherit 2 alleles for a trait, 1 allele from each parent, so they got 1 from mom, 1 from dad. The combination of alleles received by the offspring may either be homozygous or heterozygous. Homozygous, remember, homo is a prefix that means the same. And so here you see the alleles, big T, or little t, little t. Those are homozygous. And heterozygous, remember, means different. And so in this case, we have a big T and a little t. Genotypes and phenotypes. A couple more review words. Geno, genetic, kind of go together. And you'll see here the genotype. Those are the letters, the different alleles, the letter combinations that we use. Phenotype is the physical characteristics. What is it going to look like? Tall, dwarf, and such like that. Take a moment and do the vocab practice on your worksheet, and I will be here when you are finished. Welcome back. The principle of independent assortment. Mendel needed to answer one more question. When alleles are being segregated during gamete formation, does the segregation of one pair of alleles have any effect on the segregation of a different pair of alleles? So if we break that down, alleles, remember, are the different choices of your traits. Like when we have the big T, little t, during meiosis, big T would go to one of those sex cells, sperm if it's a male, egg if it's a female, and little t would go to a different one during that separation. And so these are the gametes here. Segregation would mean, are they being separated? So in this case, we're looking at with Mendel and his pea plants, if he had tall plants, would they have yellow seeds all the time? Or could he produce a tall plant with green seeds at any point in time? Were they connected or not? And so he, in other words, does the gene that determines if a pea plant is tall or dwarf have any effect on the gene for seed? Mendel designed a second set of experiments to follow 2 different genes as they pass from parent to offspring. This is known as a 2-factor cross or a dihybrid cross. Remember, di means 2. So the one parent had peas that were round and yellow, and we see these right here. And the other parent had peas that were wrinkled and green, and we see those right here. So the round and yellow traits were dominant, and because he didn't know it at first, but once he crossed them, he ended up getting all were round and yellow. So these letters here were what he was crossing. Here's the round and yellow, and here's the wrinkled and green. So if round and yellow were dominant, what's the genotype of all of the F1 offspring? So if he crossed these, what did the genotype look like with all of the babies? Think about it. Voila. And sorry it came up a little bit, it's down here a little bit, and it didn't even go all together. But if I typed it correctly, or it transferred correctly on the PowerPoint, we'd have big R, big Y, little y. Because remember, this is very similar to what we did in class when you guys had the pieces of green construction paper and you either tall plants or dwarf plants. And so here's the genotype of all of those babies. So next, he allowed these to self-pollinate and grow other plants. And here's what happened. And you can look at those. We've got, 315 round and yellow, which were the dominant and dominant traits. And then 105 round and green, which we had dominant and recessive traits. And basically what this told Mendel was that the traits were not connected. They could segregate. And so just because you had round peas did not mean they were going to be green. They could be round and yellow. So this meant that the alleles for seed shape had segregated independently of the alleles for seed color. The alleles for one gene had no effect on the alleles of another trait. This is known as independent assortment. So the principle of independent assortment states that when gametes are formed, the alleles of a gene for one trait segregate independently of the alleles for a gene of another trait. Should be, instead of principle, that should say law. Apologize for actually. Okay, so we're going to use a Punnett square for a two-factor or a dihybrid cross, and you are reading this correctly. We will need 16 squares. Each parent will pass one allele of each gene pair to the other. And so what we need to figure out, remember when we did the single Punnett squares, okay, so if we had a parent with the traits A, capital A, lowercase a, and we set up a Punnett square, we would actually put capital A here and lowercase a here. We separated them. But now we're adding in these other factors, okay? And we're going to actually have them kind of connected. Now they segregate during meiosis, so what we need to figure out with this whole thing here is how many combinations could we have? And there's going to be 4. So we're going to pretend that this is going through meiosis, and we're going to get our 4 gametes. Okay? So in other words, here's what I'm going to do. What all combinations could we get from capital A, lowercase a, capital B, lowercase b? And to do that, I would take first my capital A can go with capital B. It can also go with lowercase b. My lowercase a can go with capital B and also with lowercase b. So my combinations would end up being capital A, capital B, capital A, lowercase b, lowercase a, capital B, and lowercase a, lowercase b. Okay, this next one, we've got capital A and 2 lowercase b's. So I would have capital A with each of those lowercase b's. And I also have a lowercase a and those 2 lowercase b's. So it'd be A with a little b, A with a little b to get my combinations. Okay, same kind of thing here. So lowercase a with big B, little a with little b, little a with big B, little a with little b. Okay, while I'm writing this out, how about you try to do the other one on your own? So I would push pause if I were you. Now this next one, you've got all capital letters, so the only combinations you could have is 4 of these big A, big B's. And that is it there. Okay, so we're going to have the true breeding round in yellow with true breeding regular in green. We need the genotype of each parent and the alleles that can be passed to the offspring. So the genotype of each parent are going to be these here. Big R, big R, big Y, and little r, little y, little y. Because you see here they were true breeding round in yellow. And so that would mean they're homozygous for their traits. So to do that, remember, the only combinations we have are big R's and big Y's and little r's and little y's. So I'm going to go ahead and do my Punnett with that. I'm going to go ahead and expand it here. Okay, whoops, that was a little bit much. But here's my combinations. I would have had big R, big Y, big R, big Y, big R, big Y, big R, big Y. And then across here, little r's and little y's. Okay, now to fill in the boxes, I do the same thing as I did on a single cross, but I'm going to keep my same letters together. So I'm going to go like big R, big Y, little y. And every box right now, because all of my parental alleles are the same, all of my boxes on this one are going to be the same. So while I fill mine in, why don't you go ahead and do the same. All right, now that we have that all filled in, we need to figure out the genotypes and the phenotypes. Okay, Now you can see here every single box is a big R, big little r, big Y, little y. So what I'm going to have is big R, big Y, little y, and I have 16 out of 16 of those. So everything has that same genotype. So it's 100% of that genotype. So the phenotype with that, what the phenotype would be, since I've got a big R, we know that is going to be round, and a big Y, we know they're going to be yellow. And again, every single one is like that, so we've got 16 out of 16 like that, or 100%. Okay, and then he allowed those to self-pollinate. And so he had round and yellow, and remember, this is what we got on the last screen. And here, I actually have left, I put the punnet here for you, so you can go ahead and fill that in. I went ahead and did that, just because that's not what my focus is right now. Instead, I'm going to look at the genotypes and the phenotypes, because this is definitely a more complicated cross. So what I'm going to get with this one, let's look at the genotypes first. Okay, what I'm going to get here is in this box I have a big R and a big R and a big Y and a big Y. Now I want to look through and see how many boxes like that I have, and that's the only one. So I'm going to cross it off just so I don't come back to it. And I'm going to put that I have one. My next box is big R, big Y, little y. So I know I have that one. I have that one. And those are the only two that I see. Okay, my third box, I have big R, big Y, big Y. Let's see, I see that one, and I see that one. So again, I think I only see two. Then this box here, I've got big R, big Y, little y. And I see with that, 1, 2, 3, 4. And I think that's all I have of that one. So 4. I missed this box here, so I'm going to have big R, little y, little y. It looks like I have one. Let's see, then I've got little r, big Y, big Y. That's this box here, and it looks like I have one. Then big R, little y, little y. I've got two of that. And I've got my little r, big Y, little y. I've got two of that. And my last one, little r, little y, and I have one of that. Okay. Sorry for my sloppy writing here, guys. But I've got those genotypes, so now I can figure out the phenotypes. So big R, anytime I have a big R, I know it's going to be round. And I have a big Y, so I know it's going to be yellow. So with that, I know this one is that. I know these two are that. I know these two are that. And these 4. That's it. So with round and yellow, I actually have 9 out of 16. Okay, so then I have round and green. Oh, that was bad spelling, right? Okay, well you get it. I have round and green. So I've got this one and these 2, so that gives me 3 out of 16. And then I have wrinkled and yellow. And with wrinkled and yellow, I have that one of these two, so I have 3 out of 16. And then my wrinkled and green, I have 1 out of 16. Okay, so with that, my ratio, if I did my ratio, I would actually have 9 to 3 to 3 to 1. 9 round and yellow to 3 round and green, to 3 wrinkled and yellow, to 1 wrinkled and green. Okay, so are you ready to try one on your own? If so, you need to push pause right now. If not, follow along while the video plays and fill in your notes as well. So here's a practice problem. Right-handedness, dominant over left-handedness. The ability to roll your tongue is dominant over the inability to roll your tongue. So what might the offspring be? I have been very, very nice here and given you those. Okay, so remember the first step is to figure out how do they segregate during meiosis. And in this case, we'd have big R's, little t's. That's all we have there. And then here we have big R's and a big T and a big R and little t. So when I do my punnet here, I'm going to have, whoops, okay, big R, little t, all the way across here. Then I'm going to have, make sure I have it right, I'm going to have big R, big T, big R, little t. So big R, big T, big R, little t, going all the way across. Okay, and to fill that in, big R, big T, little t, and it looks like that's going to take all the way across the top. So while I'm filling this in, go ahead and fill your own in. Second row is going to be big R, and 2 little t's. All the way across. My third row, I'm back to big R's and big T, little t. And my fourth row, I'm back to big R's and a big and 2 little t's. Sorry. So we get to my genotypes and phenotypes. Well, across the top row on my genotypes, I have big R, big T, little t, and I can see that I've got this whole row of that and this whole row of that. So that is 8 out of 16, or 1/2. And then I also have big R, little t, and I have 8 out of 16, or 1/2 on that. So my phenotypes for those would be that, let's see, I've got R was for right-handed. It's hard for me to write on this iPad, I'm so sorry. But right-handed, and the T would be tongue roller. So we've got right-handed tongue rollers. And there would be, oh, I can't spell either. Right-handed tongue rollers, we'd have 8 out of 16. And we would also have 8 out of 16 right-handed non-tongue rollers. Okay, there's another practice problem. And guess what? This one I'm going to do completely on your own. I have set up here where I've got the traits. So copy those, do this on your own, and I will see it in class. Now here, the summary of all of his principles. Okay, so Mendel's principles form the basis of modern genetics. His principles include the following. The inheritance of traits is determined by individual units known as genes. Remember, genes is very important here. Genes are passed from parent to offspring. Each gene has 2 or more forms called alleles. That should be up in the blank. Some alleles are dominant, while other alleles are recessive. Each parent has 2 alleles for a particular trait that they inherited from their parents. They will pass 1 allele to their offspring when the alleles are segregated into gametes. The alleles for one trait segregate independently of the alleles for another trait. I do thank you for watching this. I thank you for listening. I'm hoping that it has really helped you out. And please, on your papers, if you have questions and concerns and such, be sure to write those down so we can go over them.